Now next we see that how 454 performs sequencing. So far what we have seen in the previous sessions that in the 454 the fragments are amplified in a process called as emulsion PCR, and now those beads on which those sequences are, those amplified fragments are there, they will be then processed for sequencing. So it performs sequencing using a technique which we call it as pyrosequencing, which is based upon the principle of interacting the pyrophosphate molecule, which is released when a nucleotide is incorporated into the DNA. So whenever a nucleotide is incorporated into the DNA, a pyrophosphate molecule is released, as we see here in this equation. This pyrophosphate is used to generate ATP from adenosine phosphosulfate APS molecule, and then this ATP is then allowed to react with luciferase, and that gives a light signal. So here we see there is DNA polymerase, so it is incorporating the bases. So for instance, a base cytosine is coming, so it's been incorporated, and then there is the release of pyrophosphate molecule. Then this pyrophosphate molecule, in the presence of an enzyme sulfurylase, it reacts with adenosine phosphosulfate and converts that into ATP. So that ATP then reacts with luciferase and then gives a light signal. So we incorporate one nucleotide, like for example cytosine, and then we see whether we get the light signal or we don't get the light signal. So if the light signal is coming, it means that on those particular points this nucleotide has been incorporated. So if C is incorporated, and then in the template strand there is a nucleotide G which is present, and then there is enzyme apyrase which eats up all the remaining cytosines, and then we put the next nucleotide, for example G or T or A. So in this we keep on repeating these cycles and we keep on detecting those light signals. So in pyrosequencing, only one of the four nucleotides gets into the growing chain of the DNA wherever it is complementary to the template, and then the light signal is recorded into the pyrogram. So what happens here in pyrosequencing is that after the amplification, we take those beads and which are having those amplified fragments, and we load them into the pico titer plates. That plate is loaded with one fragment at a time, and that fragment has clonally amplified copies of the same sequence. And then we also put the necessary enzymes, so there are wells in the plates which are big enough to incorporate one bead at a time. So here we see all what is happening. So we load the samples, and then in the cross section we can see that there are some wells where we have a DNA beads, and in addition to that we have enzymes in the shape of enzyme beads and also some packaging beads, so that DNA containing bead is fixed into those wells where they are sequenced. So the plate is repeatedly washed with different DNTPs and plus different other reagents, and in this way there is a fiber optic device which is coupled with the machine, and there is a CCD camera. So whenever a light signal is generated, camera takes the picture. Here we look into it in detail. So we see a bead where we have amplified copies of a same fragment, and we are just focusing on one of them. We see there is a DNA polymerase enzyme which is attached to its terminal. There is a primer to start with this amplification as well as sequencing. So whenever a nucleotide gets incorporated, there is a pyrophosphate molecule which is released, and this pyrophosphate molecule then interacts with a molecule APS, and it forms an ATP with the help of enzyme sulfurylase. This ATP then interacts with luciferin in the presence of luciferase, and then it gives us a light signal which is right here. So the light signal is then detected and recorded in the shape of a flogram, where we can see that there are these bars which are having different heights. So whenever we get a signal after incorporation of a nucleotide, we will see the higher peaks are there. So first we do calibration. We have a sequence called as TCAG, which we use it in this corner right here, and then it helps us calibration. And if we focus on just say this region where we have high peak, where we have two T's been incorporated. And then if we move on to the next one, we see a blue color. So there is a C here, again there is a T, and then again there is a G and C and G, and there are two A's. So the height of this flogram tells us how many nucleotides have been incorporated. So it can be either 1 mer, 2 mers, 3 mers, or 4 mers. So in the end, what so far we have seen here is that 454 performs sequencing with a process called as Spyro Sequencing.